Welcome to the Deep Dive. Today we're getting into something pretty fascinating, maybe even a bit revolutionary in how we think about getting older. It's called DNA methylation entropy.
That's right. It sounds technical, maybe, but the core idea is really intriguing.
Yeah, and look, we know you want the insights without wading through dense research papers for weeks. So that's our plan today. A deep dive, get you up to speed quickly but properly. We're going to unpack what this is all about.
And why it could be, well, quite significant for understanding not just how long we live, but how well we live in those later years.
Exactly. So our mission here is to explore this DNA methylation entropy idea, how it ties into aging deep down at the molecular level, and what it might mean for future anti-aging approaches. We've looked at some really interesting material, particularly an article exploring this new angle.
Should be a good discussion. Where should we start?
Well, let's start with aging itself. We see the surface stuff, right? Skin, energy, but there's this whole hidden world inside.
Mm-hmm. A sort of molecular ballet happening constantly. And researchers are digging deeper and deeper into those inner workings.
And DNA methylation entropy is like a new dancer joining that ballet.
Kind of, yeah. It's this new player that connects biology with, interestingly, a concept from physics entropy or disorder.
Okay, so before we get to the disorder part, maybe we should quickly cover DNA methylation itself. Refresh that for everyone.
Good idea. So think of your DNA as this huge, comprehensive instruction manual for building and running your body.
Got it, the blueprint.
Right. But you don't need every instruction active in every cell all the time. That's where epigenetics comes in. It's like the interpretation layer. It adds notes or highlights to the manual.
Telling the cell which pages to read and which to ignore, maybe.
Exactly. Without changing the actual letters, the DNA sequence itself. DNA methylation is one of the main ways the body does this. It's a tiny chemical tag, a methyl group, that gets attached to the DNA.
And that acts like a switch, on or off for a gene.
Pretty much, yeah. It influences whether a gene is active or silent. Now, here's the really interesting part for aging. These methylation patterns, where the tags are placed, they change as we get older.
Oh, right. And they change predictably.
Reasonably predictably, yes. Enough so that scientists have developed what they call epigenetic clocks.
I've heard of those.
Yeah.
They look at these methylation patterns to estimate your biological age, right? Not just how many birthdays you've had.
Precisely. They analyze methylation levels at specific spots in the genome, think specific page numbers in that manual, and calculate an age based on those patterns.
And that biological age might actually tell you more about your health than your chronological age.
Often, yes. It can be a better indicator of how your body is actually aging internally, the pace of it, which brings us neatly back to entropy.
Okay, entropy. I remember this from school. Something about things tending towards disorder, randomness.
That's the basic idea, a measure of chaos or lack of order in a system. Now, researchers are applying this concept to those DNA methylation patterns we just talked about.
How does that work? Are the patterns getting messy?
Essentially, yes. Instead of thinking about specific spots for the clocks, they're looking at the overall variability and, let's say, inconsistency of methylation across the entire genome.
So less about specific genes being on or off, and more about the whole system becoming noisier, more chaotic.
Exactly. The research suggests that as we age, these patterns become more erratic, less defined, the precision seems to decrease.
Okay, I'm trying to picture this, like maybe a well-organized library where the books slowly start getting put back on the wrong shelves over time. It just gets harder to find the right information.
That's a great analogy. Or like a symphony orchestra where the timing starts to drift. Instruments aren't quite in sync anymore. It's like a loss of biological information, a breakdown in that precise coordination.
And this increase in methylation disorder, this entropy that correlates with aging.
Very strongly, according to the research we've seen. Some studies, like the one highlighted in the article, suggest it might even be a more robust marker of aging than some of the existing epigenetic clocks.
Wow, okay, that feels significant. So let's connect this back. Why should someone listening care about methylation entropy? What's the potential payoff here?
Well, there are a few really exciting possibilities. First off, early detection.
Like a really early warning system.
Potentially, yes. The idea is that measurable changes in this methylation entropy might show up years, maybe even decades, before you see the typical signs of age-related problems.
So your internal disorder level could be rising even when you feel perfectly fine.
That's the hypothesis. Imagine getting a molecular heads up that things are starting to become less organized internally. That could be huge.
Definitely. What else?
Then there's intervention tracking. This is a big one. Let's say you start a new diet, exercise plans, supplements, whatever you're trying to do for healthy aging.
Yeah, you want to know if it's actually working deep down, not just making you feel a bit better?
Exactly. Right now, it's hard to measure that fundamental impact precisely. But methylation entropy could potentially be a really sensitive biomarker. You could maybe track whether your interventions are actually reducing that molecular disorder, restoring some order.
So you'd have real data showing if your efforts are kind of tightening up the ship at the cellular level.
That's the goal, yeah. It could give us a much clearer picture of what really moves the needle on biological aging.
Okay, that makes sense. And beyond tracking.
Well, there's also the potential for just deeper insight, understanding why aging happens and how certain interventions work.
Getting at the mechanism.
Right. If we see that interventions known to promote longevity also consistently reduce methylation entropy, it strengthens the idea that maintaining epigenetic order is key. It helps us understand the how and why.
So the article paints this picture maybe of a NutriTest, one that tells you not just your epigenetic age, but also your sort of internal chaos level.
That's the vision, certainly, a measure of the organization or lack thereof within your epigenome.
And that feels hopeful, actually, because if aging is partly about losing order,
Then maybe finding ways to restore or maintain that order could genuinely slow down the process. That's the underlying promise.
okay, this is really exciting stuff. But we need to ground ourselves a bit. Where does the science actually stand right now? It's confirmed, ready to go.
That's a crucial point. No, it's not quite there yet. We need to be clear, this is very much a developing area.
All early days.
Definitely. The initial findings are promising, yes, very encouraging. But, as the article points out, we need much more research. We need to validate this across different groups of people, in different tissues.
Make sure it's a reliable marker, not just something seen in one specific lab or context.
Exactly. We need large-scale, long-term studies to really confirm its robustness and its clinical utility. So lots of work still ahead.
Right. Important caveat. But the momentum is there.
Oh, absolutely. There's a lot of excitement. The prospect of being able to measure and maybe one day even modulate this fundamental aspect of aging, this biological entropy, it's very compelling.
It really taps into that deep human desire, doesn't it? Not just to live longer, but to live better, healthier.
Precisely. Extending health span, not just lifespan.
Okay, so let's try and wrap this up. For everyone listening, what's the main takeaway from this deep dive into DNA methylation entropy?
I think the core idea is that it gives us a new lens for viewing aging. It suggests aging isn't just about damage piling up or time passing. It's also about a gradual slide towards disorder, molecular confusion within the systems that control our genes.
Right, a loss of that epigenetic precision we talked about. And this new view, this entropy perspective, it holds real potential, doesn't it?
Mm-hmm. For earlier detection, for better tracking of what works, and for just fundamentally understanding the aging process more deeply.
It makes you think maybe those wrinkles or gray hairs are just the very surface signs.
Exactly. Maybe the more profound story of aging is being written in these subtle, chaotic shifts within our epigenome. It's less about time itself and more about this gradual increase in biological static, you could say.
Biological static, I like that. A kind of growing confusion that science is only now starting to really measure and understand with ideas like methylation entropy.
Well put. It's a fascinating frontier.
Absolutely. Something to definitely keep an eye on.
Ah.
Thanks for unpacking that with us today. And thanks to everyone for joining us on the deep dive. Stay curious.